Welcome everyone. Thanks for coming. I think I'll just quickly start with a little bit of housekeeping. For all those who don't know, toilets are just outside. And then if there's an emergency, we all just run erratically outside onto the oval in a single file. Just a quick acknowledgement to country. I wish to pay my respects to the Larrakia people of the traditional owners of the land we meet today. I also acknowledge all First Nations people across the lands in which we all live and work, and pay my respects to elders past and present. I also extend those respects to all of our online participants on the lands in which they meet. Just quickly, I'll introduce our guest, Andy. Andy is a research associate doing in the Population Biology and Genomics group at Curtin University. He's an adjunct here at CDU. His research interests bring together parasitology, invasion dynamics, population modelling, and evolutionary biology. These study systems include house geckos, salmon lice, fungus, farming beetles, which all bring up the questions about how humans influence ecological processes. And just quickly, we are in this room purely because upstairs is broken. We might be here for the next couple of weeks. However, hopefully that'll be fixed soon. And without further ado, Andy. Zach. Thanks, everyone. Welcome to my talk on beetles on a Friday morning. My talk, Boring Beetles and Supermodels. I was going to give you a little bit of a background about myself and how I got to this research position that I'm in, but Zach's done an amazing job introducing myself. But I am in the Population Biology and Genomics group at Curtin University. And as Zach said, I'm interested in how populations of organisms interact with the environments, how they change through space and time. And in particular, I've gotten really fascinated by parasites, how all those processes work in parasite populations, which started off with parasites in house geckos. I moved on to salmon lice for my PhD. And I was about halfway through my PhD that COVID happened. It meant all of a sudden I couldn't go into the lab. I couldn't do field work. So I was looking for a way to finish my PhD online. And that's how I got into the beautiful world of modeling. And I really came to appreciate how powerful and useful models are in exploring all these really big, large-scale processes that we couldn't possibly test empirically. And so I was creating a lot of these population and evolution models for salmon lice on salmon farms in Norway, and really started using these simulations as a kind of sandbox to play around with all of these different interesting questions, and also learned that these models are really valuable tools for actually making real-world management decisions. So in these models, I was simulating the evolution of pesticide resistance in these parasites on salmon farms. And I was looking at questions like, how can we manage different parasite controls on farms in a way that is most sustainable against evolution playing out these parasites? So I was working on this for a little bit after my PhD, but I wanted to move a little bit closer to home again. And thankfully in Australia, or unfortunately in Australia, we have a whole lot of alien invaders, a lot of destructive pest and parasite species that we can work on here in Australia. So there's about 230 or so invasive species estimated to have a significant impact on the Australian ecosystem. They are one of, if not the biggest extinction threat in Australia, certainly, and in ecosystems around the world. And so I've put up a few of the familiar faces from the NT we've seen around here, but also we have plenty of invasive plant species like ambergrass. And I feel like invertebrates kind of fly under the radar a little bit. There's a lot of very destructive invasive invertebrate species as well in Australia. And that is what I will be talking about today. We'll be talking about one of the most recent arrivals in the invasive species club in Australia, the polyphagous shot-hole borer, because that's a ridiculous name. We're going to call it PSHB for short. And this is like I said, a very new invasive species in Australia. It was first detected in 2021 in Perth. And it is a tiny little wood-boring beetle. So it's about 2 millimeters long, really, really small. They burrow into trees and leave these little tiny shot holes, hence the name. And it is a global invasive species. So its native range is in Southeast Asia. But in the last 10 to 20 years, it has spread to areas in South Africa. So it's got established populations in Israel, in California and Hawaii. There was a very recent detection in Spain, and now in Australia as well, in Perth. So it's got these established populations all around the world. And what do these beetles do? They burrow into trees and they often cause the disease and eventual death of a whole range of tree species. So in Perth, they've already cut down over 3,500 trees in metropolitan Perth. And this has been a significant loss to the community for lots of different reasons. I put up a whole bunch of news articles here mourning the loss of a lot of these really, really old, beautiful Moreton Bay figs. Trees that have quite a lot of, I guess, social importance for a lot of people living in these cities. But also Perth has the lowest tree canopy cover of any capital city in Australia, only about 22%, a little bit better in Darwin, 66%. And we know that trees are really important for people's mental health and mental well-being, but also they're really, really important for regulating temperatures, especially with climate change exacerbating the situation in cities. So if more and more trees are being lost from these major cities, that could be a huge problem for urban environments in the future. And of course, there's a lot of concerns about what would happen if PSHB spreads outside of the urban environment in Perth to other environments. They're called the polyphagous chokeholdwora. Polyphagous means they eat a lot of different species. Here's a list of 100 or so species known to be affected in Australia. The list is most likely to be a lot longer. And so that means there could be significant impacts of this beetle on agriculture. So in other parts of the world that become serious pests of avocados in particular, which is very scary. Also native forest species as well could be affected by this beetle. So I thought I'd just do a little detour into the biology of PSHB. I've been in this project for only about 6 or 7 months, so I'm quite new to this species as well. So I've gone through that process of reading the literature, learning more and more about this little critter, and I do find it pretty fascinating. It's a really interesting species, so I thought I'd share some of that enthusiasm with you all. So these beetles are a type of ambrosia beetle, and they're really cool because they are fungus farmers. So they have these specially evolved little pockets in their mouths called mycangia, and they carry around in these pockets a symbiotic fungus species, it's a fusarium fungus. And when the beetles attack a tree, they excavate this network of burrows called a gallery, and then they inoculate that tree with fungus they're carrying around. That fungus grows through the wood tissue, and that fungus is food for the whole family. So here we have another little ambrosia beetle. She's laid her eggs, and the arrows, I think, are pointing to little tufts of that fungus. When the eggs hatch, her offspring will eat that fungus. That fungus is the sole source of food for the beetle. And it's actually the fungus, not the beetle itself, that causes the disease in the trees. So the fungus blocks vascular tissue in the plant, eventually leads to its death. And interestingly, in the native range, PSHB tends to attack only dead or dying tree species. But in invaded locations, they'll attack apparently healthy trees that it seems don't have that immune defense against the fungus. So it's the healthy trees here that are attacked and heavily impacted by the fungus. And just a little bit about the life cycle of this beetle, because it's kind of important for later on in the talk. The female beetle comes along, creates a little gallery inside the tree, goes through the life cycle, egg, larvae, pupae. They emerge as adults. And I thought I'd just add a little bubble of just some really weird, funky beetle stuff that's going on here. For example, well, there's a lot of sexual dimorphism in the beetles. The females are a lot larger than the males. There's also a heavily female-biased sex ratio, so it can range anywhere from 3 females to 1 to about 10 females for every 1 male. They have a haplodiploid sex determination system, which means that females can produce males asexually. And most interestingly, they are obligate inbreeders. So in most species, we assume that inbreeding has a lot of deleterious impacts on the population. But for this species, they mate almost exclusively within their own family. So inside the burrow, the siblings will mate, the females will then emerge. If there's no males around, the female can produce a son, which she will then mate with. They're all a little bit weird, but they also make the beetle a really, really successful invader, because all of these together means that a single female can arrive in a location and produce an entire invasive population without needing to find a mate, without having any founder effects impacting the genetic quality of that population. So the female will then start a new gallery in the same tree, or she will fly off a short distance to a new host. Okay, so what's being done about this invasive species? The WA government has put forward a $41 million containment and eradication plan. There's a lot of monitoring set up. If you go to Perth, you'll see these little sticky traps plastered all over the city. The idea is to catch those female adults as she's flying between trees, to try and figure out where these beetles are spread to. But at this stage, there's only one treatment against the beetle, and that is to either chop down the tree or, at best case, chop down the infested branches. So it's not an ideal scenario. There are some quarantine zones set up. Initially, there was this little blue quarantine zone set up around, reminds me of COVID, actually. Reminds me COVID. Yeah, the quarantine zone where you were prohibited from transporting timber between inside and outside the quarantine zone, because these beetles live inside timber and they're really small. It's really easy to accidentally transport them to new areas. However, in September 2024, they expanded that into the red zone, which kind of speaks to the difficulty in trying to contain this outbreak. And there's also some pesticide trials being trialed as well, but all very early days at the moment. So given all this, we really want to try and manage this new invasive species as effectively and efficiently as possible. So we want to be asking questions like, how far could PSH be spread? Which areas are most at risk? That could be which areas are most at risk of being colonized, or which areas are most expected to have the biggest impact if that area is colonized. And then we want to try and manage our control efforts most effectively. So this means those monitoring efforts I talked about before. We want to try and monitor in places where we have the highest probability of detection success. So we want to know that the beetles are there if they're actually there. And we want to be able to control the beetles most effectively. We need to be strategic about this. We can't put a lot of effort into controlling beetles in one area only for that to be colonized shortly thereafter. So there's a lot of moving parts here. We need to try and coordinate and optimize the strategies that are available to us. And this is where models can help us out. So today I'm going to be talking about species distribution models, which is a broad church of modeling processes. And the general idea here is to map the potential range of a species across a landscape according to the suitability of different environments for that species to live in. So these are widely used in a whole range of ecological questions, but they are really interesting and useful for invasive species in particular. So for an example, here's an SDM of cane toads in Australia. This solid black line is was the current range of cane toads. It's now, I think, spread almost to broom. But then this shaded area in Australia shows the potential range where the cane toad could actually spread to, given the suitability of that environment. And in particular, if we zoom in over here, we see this very, very narrow range of, narrow corridor of suitable environment through which the toads could spread from Broome into the Pilbara, which would be a big problem. And it's this area that's the target focus of the toad containment zone, which is a really, really cool project that my supervisor is working on in WA. I won't go into it now, but it's illustrating how SDMs can really provide insights into how invasive species can spread through space, through time. And just another example here is the predicted distribution of this invasive willow species in the Bogong High Plains in Alpine Victoria. And can't see it super well in this plot, but the shaded areas show all the places where this invasive willow is expected to be able to persist. But in managing this invasive species, people actually have to go out to this really difficult country and invest a lot of time and effort into finding and removing these species. And you couldn't possibly do this across this whole area. So what the authors of this paper did, they created this species distribution model, but then fed this through another decision-making optimization model, which essentially said, where should we prioritize our control efforts? So if we only focus our efforts on the places where we have the highest chance of detecting this species, where control efforts are going to have a higher success, and also prioritizing areas that are most at risk of this species, these are the little spots, and maybe you can't see it super well, but these are the little spots where our management efforts should be concentrated. So lots of really cool, interesting things you can do with these models. And the SDMs are kind of the foundation upon which you can build additional management strategies. A little bit of technical stuff. Broadly, there's two types of species distribution models, correlative versus mechanistic. So correlative models, you take occupancy data, so presence, absence data of a species that's known in an area, you correlate that with environmental predictions, and then you extrapolate that onto an environment. Say, where could this species live given its known occurrences? So there's a lot of really useful packages available for this. This is a really widely used modeling system. It's versatile, it's efficient. You can use it for a whole range of taxa. You don't need a whole lot of new data. A lot of this data is readily available for people. But the crux is that there are a few assumptions to the correlative approach. In particular, they're more prone to error when you're extrapolating into completely new environments, and also if you're dealing with populations that are not at equilibrium, so populations that are spreading. And these two things are obviously hallmarks of invasive populations. So we're actually turning towards the mechanistic approach instead, which is a lot more of a robust system for predicting invasions of species because of these assumptions here. So the mechanistic approach, you take data such as physiological data, and you essentially predict the fundamental niche of a species. So you're saying, given the physiological processes of this species, where could it, within the realms of its physiology, actually survive in an environment? This will make more sense as I discuss it a little bit further. Be looking at the fitness of a species given environmental conditions. These models are more demanding to develop, however, they take a little bit more time. But there are benefits to this process as well. You can explore a lot of factors that determine the distribution. So the correlative approach gives you a general relative suitability of different areas. This place is more suitable than this place. But the mechanistic approach lets us ask, okay, why is this place not suitable for this species? Why is this area highly suitable? How can we exploit that in our management efforts? And also the mechanistic approach is often linked to a population model. So rather than looking at the fitness of an individual in a population, in a given environment, we can look at the fitness of an entire population. And that lets us learn more about invasion dynamics. And again, this is the process that I'm using for the PSHB. So this is a little crash course in SDM theory, but we're going to go into more detail now. By starting off, we're going to create a population model for polyphagous shothole borer. That's fun. So it's a relatively simple stage-structured matrix model. It looks kind of like this. Essentially, you're calculating the number of beetles every day over the course of a year. And I'm very aware that a whole talk with maths equations is not very fun or exciting for people. So we're going to take this equation and kind of shift it around a little bit and make it a little bit more intuitive for us all to understand. So essentially the same kind of equation. Now we're splitting the beetle into three different components. The adults, the juveniles, we've condensed all the juvenile stages. The pre-ovipositing stage. So these are the females that are adults but haven't yet started a colony. This is our little life cycle. So every day, adults produce some new offspring of fecundity. Every day, some of those juveniles transition or develop into the next stage. The pre-ovipositing stage transitions into the reproductive adult. There's our little life cycle. We also have a background mortality rate. So every day, a small proportion of these individuals die of natural causes. And we also have a proportion of these pre-egg laying females dispersing away from our population. But we assume the immigration rate is the same as the immigration rate, minus some mortality of those beetles. We assume that some of those beetles die en route between different hosts. But this little bit on the left-hand side, we're just going to ignore for now. We'll push that to the side. We'll focus on it later. We're just interested in a single population of PSHB, which is given by our model. The last consideration is, and this is really important, that these beetles are ectotherms, so a lot of their processes are dependent on temperature. So depending on the temperature, the rate of development and also the survival rate, mortality rate of juveniles is going to differ. So that's our model, but we need values for these parameters. And this is one of the hurdles that we come to if we're using that mechanistic species distribution model. We actually need to know a lot about the life history and the life cycle of the species to actually construct our model. Thankfully for us, there's already really good life history on this species and its closely related species from lab studies. We've got a few studies here. They monitored the life cycle of this beetle in little test tubes at different temperatures. And this gave us a whole stack of data that we then plugged into that model I had at the start. And the most interesting maybe are these little data points here. So from these papers, we've got the transition probability again. So that's the development rate and the survival probability of those juveniles across different temperatures. We fit these thermal performance curves to these data points. And this gives us these really nice curves. It illustrates what I was talking about before. As temperature increases, the development rate and survival of these beetles increases up to a certain point, which is our thermal optimum. And then beyond these temperatures, the fitness then declines again. And this is really captures what I was talking about before with the fundamental niche of a species given its physiology. We know that the fitness of these beetles are really going to be best within these little pockets here of high fitness at optimal temperatures. Okay, so we've got a population of PSHB. We're modeling that through time. This can either be in a single host or it can be in a cluster of hosts. It doesn't really matter. It's the beauty of models. You can make a lot of assumptions here. And we want to model the effects of daily temperatures over 1 year on a population. So it's one thing to just model this population at a single constant temperature, but what we're interested in how this population will react as we go through the seasons, temperatures get colder, the temperatures get warmer again. How will that affect an actual population of beetles? And to do this, we used the Silo Climate Database, which is a really fantastic database that I've just started using for this project. It's run by the Queensland government, and it is called, what does it say? Climate data from 1889 to yesterday. So you can get climate data, historical climate data from over 100 years ago on any given day. And they use an interpolation method for the weather stations, which means you can estimate the climate data for any given location in Australia as well as any given time. It's a really, really powerful resource for us to use. For example, here's the maximum temperature in Perth over the last 5 years from this silo system. Okay, so we take all of that climate data for a location, in this case Perth. We put that into our model. We put all of our beetle life history parameters into our model, and we're just going to run it. So here's one of the outputs we get. It's the daily temperature, mean temperature for Perth. Looks like that. And this is our population of PSHB. So we started the model with just a single individual female, a foundress. And as we see, there's a pretty clear exponential growth of this population. We didn't have any carrying capacity in this model, but that's something we can add later. But if we log transform that, so we see the population continues to grow in Perth through time, but there is a little bit of a dip here over winter. The temperatures are colder, the population doesn't grow as quickly. And it's the slope of this bottom graph here, the growth rate is what we're really interested in. You can plot it like this. This is the rate of population growth, PSHB, through time. Obviously, 0 means the population is static, no growth. Negative means the population is actually declining. And what this tells us is that from a single adult female beetle, she will continue to grow a population over the course of an entire year. But the rate at which that population will grow will slow over winter and then increase again over summer. This kind of makes sense for an ectoderm. But it's this plot here, or this value, the population growth rate, that we can use as a metric of habitat suitability. The faster the population grows in an environment, the more suitable that habitat is expected to be. We can then use that to create our species distribution model. So we run that same model I just described, but across a 10 by 10, across a grid covering all of Australia, the resolution of 10K by 10K. And we're essentially mapping the variation in that population growth through space. And I did promise supermodels for everyone. Mine is not so glamorous. Unfortunately, I've been running these models on the Cetonix supercomputer at the Pawsey Supercomputing Research Centre, which is another really valuable resource that I've had available. This model isn't the most computationally intensive model out there, but each simulation creates, I think, 150 million data points or so. So it's really nice not running that on my laptop. So thanks to the team there, the supercomputing. And here's, I guess, the output. And one of the beautiful things about modeling, in my opinion, is that you get to create these really nice maps that just look kind of cool. I think they look kind of cool anyway. And this is showing the relative suitability of environments across all of Australia for PSHB. So we see here, down around Perth is a pretty high suitability area. It's no surprise that the beetles, when they arrived there, took off really well. We see there's a little bit of suitability around here, down south in SA. I feel like a weather person, actually. And then up along here, Queensland, there's a really, really high suitability. So what this is telling is, without proper biosecurity controls, if PSHB could make it to Queensland, in particular, it could have quite a catastrophic effect on the environment, particularly given a lot of the food production, avocados in particular. So this highlights, how many different areas in Australia are most at risk. Conversely, high altitude or very cold environments such as down in Tassie are going to be not very suitable for this beetle. A few things to note. This is showing us the fundamental thermal niche of this beetle, which areas have the best thermal environment. Of course, we haven't included a whole range of other factors in the species distribution model. We haven't got any other abiotic factors other than temperature. But we think because these beetles spend most of their time ensconced away in the wood, they're probably not directly affected by other environmental variables so much. However, of course, the other main thing we haven't included are biotic factors such as host availability. Most notably, maybe there's a really high suitability environment here in the Nullarbor, not many trees in the Nullarbor for these beetles to live in. So this model is essentially telling us if a beetle could affect a host in this area, how well would they go? But if we were to overlay host availability to this map as well, we would probably shrink this map or shrink the suitability of the map into a more realized niche. And that's something we're planning to do further down the line. So what some people in California did, I won't go into too much detail, but they had a thermal suitability map, which is the greens and the blues. But in little locations, they also had vegetation data, and they mapped out the density of host species for these beetles to attack. And so it really kind of shrinks down where they could, it shrinks down, the thermal suitability into a more realized or more realistic range of species. But you do need to have good vegetation data. And one of the issues is that these beetles really like to attack a lot of exotic ornamental species. And there's not a lot of good data on the distribution of these ornamental exotics because people will have them in the backyard without anyone really documenting that. So the range is probably quite high, relatively high suitable range. I thought I'd zoom in quickly into Southwest WA. This blue line is that quarantine zone I mentioned before. They were probably introduced by boat around here, Fremantle. And there's a lot more work we can do to really delve into this results in greater detail. But straight off the bat, we can see that the suitability down south is not so high. But if the beetles are going to spread anywhere, it's most likely going to be up north along the coast. So potentially a lot of control efforts should be focusing on these regions along the coast further north, because these are the high risk areas for the beetle to spread to and have high impact. And also remember that we're looking at these results through time as well as through space. And this can be really interesting or useful for monitoring efforts as well. So remember, the monitoring usually takes place with those sticky traps. We're catching the female beetles as they're flying between trees. And we have a higher chance of detecting those females during periods of high population growth, when we expect there to be lots of reproduction going on, a lot of reproductively active females flying around. And so what this means, maybe the best time to set up these sticky traps is around here, after a long period of population growth, high population growth, before that winter lull. That gives us the highest chance of detecting them if they're actually present. I thought I'd just introduce or include a little bit about NT, bit of Darwin as well. They do pretty well in Tiwi and East Arnhem, but not so much around Darwin. As you can see from this spot here, they're really happy during the dry season. But once we get into the build-up, it's actually getting too warm for them, and the population can actually decline a little bit over those extreme conditions. Okay, there's a few other little things we can do with this model though. One of them is in estimating invasion speed. So these are all the little things we can do with the mechanistic approach that I talked about before, and particularly having a population model built into our SDM, we can estimate invasion speed. Because in an idealized scenario for a population at equilibrium, invasions spread at a constant rate. You can see this in Petri dish with bacteria or fungus. If you put a little drop in the middle, it will spread across the Petri dish at a constant speed. And we can actually estimate that speed with this little equation here. There are two components to it: population growth, which we've gotten from our model, and also coefficient of diffusion or dispersal. So this is another study, actually didn't include reference to that one. This was a mark release recapture study with these beetles. And they released them at a little point here, bird's eye view of an orchard, and they caught the majority of beetles within about 20 to 30 meters away. So we can take this data, plug that into the equation as well. And what this tells us is that the beetles could spread at an upper limit of only about 4 kilometers per year, which is pretty fast for a little beetle, but in terms of invasion, pretty slow. I think cane toads spread at 50 k's a year or so. So it's a pretty small spread rate, which is quite comforting for us to know. But these beetles are most likely to spread through human-mediated transport, the transport of firewood and tree clippings and things like that. So that's the most likely route then for these beetles to invade new areas, not from their self-driven dispersal. And finally, we're just going to go a little supermodel world tour. We can use this model for any location in the world using the NASA power project. This is kind of like the silo climate data I had before. It's climate data for any location around the world. And what we wanted to do, we wanted to run our model for already invaded locations, places where we know there are already established PSHP populations, for a few reasons, mainly to make sure the model's actually giving us reasonable results, make sure it's working how we expect it to work. So for example, we know there's a population down here in California, and this is our model for California. As you can see, the area where the beetle is present is the highest suitability area. And generally speaking, there's positive population growth around the year. If these plots were below 0 for long periods, that would be saying beetles can't live here. It's too hot or it's too cold for beetles to live. And we know the beetles are there. So that would be a bit of a problem. But the fact that our model is saying, yes, beetles are happy to live here, that's kind of giving us a bit of validation that the model is working relatively well. And also South Africa as well. We have a few distinct populations across South Africa. We ran our model for South Africa as well. Once again, the model results seem to be pretty good. The locations where the beetles are present are places where our model's saying it's good habitat. Notably, this little area up here is really, really high suitability, but there haven't been many records here, so that's interesting. It could speak to a lack of monitoring effort in those areas, though, or the beetles just haven't made it to those parts of South Africa yet. And one last little thing, that we can use the model to estimate uncertain parameters. For example, if you cast your mind back to my discussion earlier with the life cycle model of the SHB, we talked about the dispersal rates. So these pre-ovipositing females dispersing to and from a host tree, and there's some mortality that goes on during that dispersal process that we just don't know. As I said, there's lots of great data in the literature for this species, but it's really, really hard to measure dispersal mortality in this species empirically in the lab and field. So we don't know what this value is, but thankfully we can use the model and the fact that we have known locations for this invasive species to actually try and estimate a ballpark figure for this mortality rate. And so what we did, we ran a whole bunch of simulations with that parameter for dispersal mortality ranging from 0%, so no beetles die when they move between trees, to 95%. And then we looked at the mean population growth rate, that population, and we ran that for a whole range of locations where there are known established populations of PSHB. So we get a plot that looks kind of like this. As you expect, the higher the mortality rate in our model, the slower the population growth rate. And eventually we get to a point where the population can't persist, growth rate becomes 0 or less than 0. But we know that there are beetles in these locations here. So this parameter, assuming all else in our model is relatively correct, this parameter here must actually be less than about 40%. So this gives us kind of an upper estimate of what this parameter might actually be. And now we can run the model again with this new parameter for dispersal mortality and see how that changes our results. So if we ran that for Australia again, now we have 40% mortality of beetles as they move between trees. That's going to change our population growth rate. And here I've just included places where population growth is greater than 0, where populations can actually persist. And yeah, it very fundamentally changes what the map looks like. Now we see there's just this continuous band of habitat running from the southwest of WA down south and all the way up to Queensland. And this is where we might expect these beetles to survive given this new parameter. So this is all a work in progress. We can really just use the model and play around and tinker with different values to try and get a better understanding of how this species might invade new areas. And so, yeah, some future directions. Like I said, I've only been doing this for the last few months. There's a lot more exciting stuff we can do with this model. We want to add some monitoring data, deeper in WA, have quite a bit of monitoring data available, knowing the presence and absence of PSHB in different locations. If we can work with them, we can incorporate that into our model, and it would really just bump up the power of this model and let us ask a whole range of new questions. Because of the matrix population model structure that I've been using, it's really modular. I found this with my salmon lice model previously. You can just keep building and adding new elements to this model quite easily to explore a whole range of really cool new things. For example, we can simulate the dispersal of different populations. So you can start off with one invaded location and then watch how that infestation spreads across the landscape through time. We can incorporate controls into our model. We can say what happens if we treat this area, what happens if we treat this area, how will it affect the populations. We can overlay host availability, as I mentioned before, onto the model as well to really contract where we think the species might be able to infest. And all of these ideas further down the line are really aimed at asking this main question is how do we coordinate management efforts most effectively based on our results? So how can we really assist DPIRD and assist all of these control and monitoring efforts most efficiently and most strategically so that we can have the best shot at containing PSHB and stopping it from spreading to new locations. That's all. Thanks very much for sitting with that all. Yeah. Anyone have any questions for either online or in the room?
Thanks, that was a great talk. I was just curious about, have you or anyone done any modelling of the fungus?
No, that's a great question. Not that I'm aware of. That's a whole other side, because of course the fungus will have its own thermal profile, its own physiology. For this model, because the beetle needs the fungus to survive, in all those lab studies, it was feeding on that fungus as well. So all of the data we have is kind of implicitly for the fungus and for that beetle itself. If the conditions were not so good for the fungus, the beetle would lose its fitness as well through time. So yeah, all our data is kind of assuming this mutualistic relationship between beetle. But that's a whole other fascinating world, because these species do pick up new strains of fungus. They do, they host switch these fungus species. It's very possible that in Australia it might pick up new strains or species of fungus to use as well, which would completely, change its potential impact on the environment. So no, I haven't, but it would be very interesting. Yeah. Oh, Anna at the back.
Yeah, this is from Keith online. He's wondering, how different are the temperatures inside the wood from the mean air temperature outside?
Oh, fantastic question. Here's a slide I prepared earlier, potentially. Oh, I have to go through all my. Oh no, how embarrassing. All right, don't worry. I did have some slides somewhere. I was going to include it in the talk. I was worried about running over time. But yeah, absolutely. These beetles are living inside the wood. The temperature profile is probably quite different. Or there are some studies that show that the temperature inside sapwood is a few degrees different than the ambient temperature, which is of course going to affect our results. So we have actually accounted for that. We created a model, function that takes a lot of environmental variables and then predicts the average internal tree temperature. And that's actually the temperature that we used to predict PSHB fitness. A great question. But we do have a very limited data set. We've been asking around a whole bunch of plant physiologists trying to find any data sets for internal tree temperatures. We're quite limited. If anyone does know of anyone or have like data sets available, that would be actually really, really helpful. Because it's not a thing I think people tend to set out to measure. But for in cases like this, it's actually really, really useful. Yes, so I have accounted for that, but would love more data to really make sure that function we have is as accurate as possible. Oh yeah, well, yeah, I was gonna. There we go.
The weird inbreeding thing. Is there much info about whether they do that at the same rate they get away with it somewhere new where the trees are all naive, or did they do it right across the board?
I think they do it right across the board, because those males, most of the time, the males never leave their little gallery that they're born in, so they don't disperse. And the females will always move to a new gallery to start a new one. So, yeah, they seem pretty sort of honed in on that strategy. And it's, I guess the theory is, from my very limited reading, is, for whatever reason, generally inbreeding is bad for a population, because you're kind of accumulating a lot of these deleterious genes that affect your fitness. But at whatever point in history, these beetles have managed to essentially purge themselves of all these deleterious genes. So essentially, they have only the good ones that they need to survive. And that has allowed them to persist with this strategy. It does mean that genetic diversity is probably really, really low. And I wonder, in terms of like biological control, that can be a really interesting strategy. Yeah.
Just a comment. I think I heard Ben cringe from Perth when you said dispersal rate. We can assume it's constant through space.
Oh, true. That's a fantastic point as well. That's a very, very big blanket statement. The constant dispersal that gives us like an upper ballpark estimate, but of course the rate of dispersal is going to depend on the environment you're in, going to affect population dynamics and stuff.
So Perth being the least treed capital city in Australia, presumably that's partly because the habitat is sand plain and trees aren't abundant there. It's a good place for them to arrive potentially. Is it mostly in planted trees that they're currently invading?
Yeah. So in that list I had before, the top 10 heavily infected tree species that are kind of reservoirs for this parasite are all exotic. You have like plane trees, poincianas, a lot of not native species, but because they're heavily planted around the urban environment, that's propagating. As for why Perth has such low tree canopy cover, I'm not sure. That might be. I'm also hoping it might just be like a very urbanized, wide streets.
Yeah.
I do wonder though that makes it actually, it probably would have slowed down the rate of dispersal for sure.
Comment as well. It's a real failure of biosecurity. Anyone who's trying to come back through customs to Australia, I thought wooden products were really scrutinised.
Yeah. I mean, no one knows exactly where or how it came in. As I think I mentioned, because they're so small and a single beetle in a piece of wood and a single female can obviously produce this entire, vibrant population. But yeah, it slipped through the cracks somehow, and now, unfortunately, the government's really got to sort of deal with it. Yeah.
I tried to backtrack. I was going to say, knowing the rate of increase as to the actual year when it came in.
Yeah.
It's only been around for a long time, and it's only just got to this point
That's-
where the numbers are high.
Exactly, very, very true. And that's one of those things that if we have presence absence data, we can actually use that to backtrack and estimate the arrival date. At this stage, we don't really know. It was first detected around Freo Port, and so kind of suggests that it probably came in by ship. But again, we don't really know. Could have been there for a while for sure.
Question around that. Just a question around that, like how wood products are really restricted to bring into Australia. And if it's even a little piece of wood, how long do these beetles or fungus survive in that small piece of wood?
Yeah, great question. I mean, in a tree, they can definitely survive for months at a time at cooler temperatures. Again, it's temperature dependent. I'm thinking they have done studies where they'll leave pieces of wood out, and usually the females will crawl. They can, I guess, tell the wood that they're in is cut up and dead, and they will actually emerge. But yeah, it might be a few weeks potentially at a time. It depends on the temperature. In Perth, they suggest leaving if you've got any wood clippings, cut down trees, chop it up into small pieces, wrap it in a plastic bag, and leave it in the sun. And that'll kill them, in theory, relatively quickly. But yeah, on the ship, who knows what kind of conditions it was in? Not sure. Yeah.
We have a few more online, following on from that one actually. Adam is wondering, what's the lifespan of a single individual? And another quick one here, Carlos is wondering, are the burrows hard to spot when examining trees?
Yeah. So the second question first. Those holes, those little shot holes, are really, really small. I think there's horrific slides here. There we go. About the size of like a biro point. So if you know what to look for, they're quite distinctive, but they are tiny, and unless the lumber's covered with them, it's probably quite easy to miss one. How long do they live for? I don't think there's any studies actually measuring that, unfortunately. But in cooler temperatures, like I said, months at a time. They think in temperate climates, they can overwinter, so they can hide in the tree and just survive over winter. So probably a few months at least, potentially longer. But we don't really know. Any other questions? Yeah, go.
Depending on how the climate here is in Darwin, like the weather is quite warm, and we don't have much coast, but how likely would you think if certain piece of wood comes here in Northern Territory, how likely is it to establish here?
Yeah, I mean, there's a lot of I don't know in this whole thing because it is such a recent thing. There's not a whole heap of research being done, and the research that has been done is not really based on the Australian climate. But probably pretty well. They're native to Southeast Asia, sort of Indonesia, Thailand, south of China. So it's probably pretty similar climate. That map I had up before said around Darwin is maybe not so suitable because the temperature inside those trees, they might just cook inside those trees over the buildup. But again, different tree species have different like thermal properties in the wood. It could be the trees up here are more thermally resistant or able to better insulate themselves against heat. So maybe beetles could survive up here. Yeah, our models are saying it's a possibility. It's not a bad place necessarily for them to live. But whether they could sort of spread outside of Darwin into the natural environment is a different question.
Two quick questions. One, obviously Australia is somewhere that gets burnt a lot by fire. Has been any research into how fire can affect spread or have I guess, a positive impact in terms of security?
That's a fantastic question. Yeah, I don't think that has been at all, but that would be a really interesting question.
And so
I assume fire is probably not very good for them, but if it's a tree that burns really quickly and leaves the inner like candle tissue intact, then maybe the beetle's quite happy. It's cooking in there for a little bit, emerge after. So.
and to throw you under the bus, something that Andy said to me is all models are wrong, but some are more wrong than others. What's the accuracy? Like how reliant? Like is there like an error of the model? Like how confident are you?
Thanks for that. It's one of those things that the accuracy is only going to improve once we can validate it against data. So this is why, getting monitoring data for Perth is going to be really, really valuable to actually fine tune and hone our results. Being able to kind of compare our models against what we see in like California, Israel, South Africa helps our confidence a little bit. We're fairly sure that those life history parameters are pretty accurate, but how that translates into the actual environment, we don't know. If nothing else, a lot of those maps just give us a relative suitability. So we might not know the exact suitability of one location over another in terms of population growth, but we do know that this place is better than that place. Work from there.
We've got time for one, maybe two more questions.
So the fire question actually brings up a good point, right? We know from studying animals in fires that shelter in tree cavities, they're extremely good temperature buffers. So that, the fires are absolutely raging around the tree, but this really temperature sensitive animal persists because they're buffered from extreme temperatures through inside the tree. So maybe the temperatures in the trees are like really quite different to the ambient temperatures.
Yeah.
And maybe that's an opportunity to get some of those internal temperature data. So like it would come from tree cavities, which is not the same thing.
That's the thing we have thought about tree cavity data, which has a whole heap of great data, but I think that is quite different. Tree hollows, really beautiful buffers, generally a lot cooler. I think actual inside the living wood is quite a bit different. If we have more data to work with, that might be something to explore. Because if we can just sub in tree hollow data, that's great.
How would you get the internal temperature of the tree? You have to bore into it, right?
Yeah. So what the data we do have comes from sap flow loggers. So little probes they put in the tree, and it's to measure the rate of sap movement in trees. But they incidentally measure the tree temperature as well. And there's a whole bunch of people have studied sap flow in trees across different environments. There's a whole like online database. But either those loggers don't record the temperature, or the people doing the research just chucks that temperature data out because it's not useful for them. So it's quite frustrating that there's probably all of this data being measured, but we don't actually have access to it. That has crossed my mind, and that would mean, I guess, yes.
$41 million. You got a bit of money.
Yeah, true.
This would be the last one from online. It's from Gatawan. He would like to know, does the model fit with multiple environments?
And what's the second one?
Absence.
Absence data. Yes. Well, we can absolutely add more environmental variables to it. Yeah, as I said earlier, I think we probably don't think other environmental data and environmental variables are going to directly affect the beetle because they're in that tree. But if they're related to host survival or host density, that's absolutely going to have an effect. So yeah, there's definitely room to include a whole bunch of different variables in there. We just need to get that data and absence data as well. Yeah, that's sort of tied in with the monitoring presence absence data that we can get from the government. Because yeah, it's really important to know where these beetles are, but also where the beetles aren't is really, really important. As long as we can, obviously there's a detection probability bias there, so we need to estimate that as well. Yeah.
So there's morning tea in yellow too. So if anyone would like to join for morning tea, chat more to Andy or other real people, please feel free to go up to the top of yellow two into the kitchen. And
If you don't want to chat to Andy, Maurizio, the new director of the RI, is coming. You could talk to Andy and me, Maurizio.
yes, thanks everyone.